right, so our next presenter is Fred Misowicz, Chief Operating Officer of Argos Therapeutics. Piper rates Argos overweight with the $18 price target.
Okay, thank you for the introduction. We went public in February of this year, so I'll show you this slide on forward-looking statements, risk and uncertainties. And what I want to do is discuss the therapy itself, show you our clinical results, and also talk about how we plan on commercializing this technology. Arcellus is the name of the platform that we call this technology. It's fully autologous, and we use RNA from the patient's tumor, which represents all the self-antigens as well as mutated antigens in that particular tumor. And we use the dendritic cell, and we put that messenger RNA from the tumor in the dendritic cell. We express those mutated antigens, and we get a very strong memory T-cell response. And this T-cell response has been shown to correlate with clinical outcomes, and I'll show you some data on that in a few slides. We've also have prototypes where we've automated this process. It's very manual, labor-intensive, and one cannot commercialize an autologous therapy using totally manual methods. And I'll discuss the automation of this process as well. Currently, we're in a phase 3 pivotal study in metastatic renal cell carcinoma under an SPA, which for some of you, that's called a special protocol assessment with the FDA. They look at your clinical trial design, your endpoints, your magnitude of clinical efficacy, as well as your analysis plan. And while it's not a guarantee for approval, you won't get rejected on your BLA when you file it. In our phase 2 study, which was in combination with Sutent, which is a first-line therapy for metastatic renal cell, we demonstrated a median overall survival 2 times greater than Sutent alone, and long-term survival greater than 40 months. Typically in this patient population, only about 5% of the patients live longer than 30 months. And we also have completed a phase 2B randomized placebo-controlled study in HIV. We call this AGS-004. This has been fully funded by a contract of $39 million from the NIH, the AIDS division. We expect to announce that data mid-2014. And I'll talk about 2 trials that we're planning for later this year that involve eradication of the virus or elimination of retroviral therapy. So let me talk to you about the design of the therapy and what we think the significant attributes are for a good, effective autologous immunotherapy. First of all, we believe that the immune system really needs to see the mutated antigens which are present in that tumor. There's been no successful immunotherapy just looking at overexpressed normal antigens, so that's part of our immunophilosophy. Secondly, these patients are profoundly immunosuppressed. I think you've heard that in earlier talks, and this exists both in cancer and infectious diseases. And our proprietary process to produce the dendritic cell overcomes some of this immunosuppression. Just as an example, in our first generation therapy, these patients, T cells can produce IL-2, but not gamma interferon. In our current process, they can produce both cytokines and act more like normal unsuppressed cells. We also induce, as part of our mechanism of action, a memory T cell. And I think you've heard about memory T cells today when you heard the CAR T cell talks. And this is a CD8, CD28 positive memory T cell, and it is not susceptible to PD-1 checkpoint inhibition. Since the therapeutic product is fully autologous, we have minimal toxicity. I think you've heard that in earlier talks. Small injection site reactions, typically grade 1 or 2, some swelling of the lymph nodes, but all are transient and resolve. And then finally, and I think just as important as clinical efficacy, is we have a cost-effective automated system. It's scalable, consistent. One can roll it out from indications, medium size, like metastatic renal cell of about 10,000 patients, up to HIV, 50 to 60,000 patients. And since it's a platform, basically the cellular process of making the dendritic cell is the same for any cancer and any infectious disease. And for solid tumors, basically the same process will work on a variety of solid tumors. HIV is a little bit different. I thought I had to show this slide to this audience, but after hearing the panels this morning, I don't think I do. But Big Pharma has taken a large focus on cancer immunotherapy. We've heard about Novartis and the CAR T cells. Pfizer announced collaboration with MD Anderson on novel immunotherapies. And, we feel this is a good sign for us smaller biotechs that have been in this field for over 10 years. What I want to do now is show you some clinical data on AGS-003 and metastatic renal cell, and I'll show you the results of two small single-arm trials, not controlled, but still gave us a clinical signal that we think is important. And in this disease, and I don't want to go into all the details, the patients are stratified by risk factors. It's heterogeneous. In other words, favorable, intermediate, and poor. And you'll see, as I show you the data, that all our patients are either intermediate or poor risk patients. To be favorable risk, we're diagnosing the patients just as they get the disease, and they're treated in less than a year. So that automatically puts them either in the intermediate class or lower, depending on various laboratory tests we do. But really getting back to the data, we did a single agent study shown here on 22 patients, looking at overall survival as the primary endpoint, and compared that, and this is historical data on Sutent, which is first line, as I mentioned, and a tyrosine kinase inhibitor with a database of almost 1,200 patients. And as you can see, we're about the same in overall survival, slightly less favored in terms of having more poor risk patients, which have a poorer outcome in terms of survival. When we combine Sutent along with our therapeutic autologous therapy, we saw a doubling in median overall survival up to 30 months. We saw that the intermediate risk patients alone had a median survival of 57 months, and basically also observed that the progression-free survival was twofold over that with, let's say, similar patients in historical database with Sutent alone. And I'll talk about our phase 3 design, but really we're designing, or had designed our phase 3 to really mimic this distribution of intermediate and poor risk patients, with favoring intermediate slightly more than the poor risk in the slide. Going to long-term survival, and I mentioned that we had a fourfold increase in long-term survival over comparable patients. The single agent showed that 23% of the patients live longer than 30 months. Again, in combination with Sutent, we saw this dramatically increase to 52%. For the single agent trial, given the median, we all had 5 subjects that really had long-term survival, almost up to 4 years. And in the combination study, a third of our patients survived for 54 or 4 months, which is, historically, this is way beyond what's been observed. Now, coming back to this CD8, CD28 memory T cell, this slide shows the correlation between the immune response, and by immune response, we're looking at an increase of tumor-specific memory T cells before and after the 5th dose. Typically, we dose the therapeutic at 3-week intervals, and it takes about 5 doses to get the immune system fully activated, let's say, against the tumor. So the top part of this graph shows the increase in these memory T cells. The bottom part, and correlates directly with the patient, shows the overall survival. And you can see that with a P of 0.8, P less than 0.002, and Spearman's coefficient, this is a significant correlation of a cell that we observed in the patients during these studies, albeit small, that correlates with outcome. This cell also correlates with the doubling of progression-free survival, and also correlates with tumor response. So in our phase 3 study, we're going to have 300 patients that we can observe to see if this correlation holds up. Now, this is the design of the phase 3 study. We started it in 2013. It's 450 newly diagnosed, as I mentioned, intermediate and poor risk advanced cancer patients. It's controlled, randomized, open-labeled. Had a similar design to the phase 2. One important exception is in discussions with FDA. An FDA on an SPA is a great thing to do because you get a lot of good advice both on your clinical trial design as well as on your CMC section. And basically our argument was that since this therapy is not toxic, we want to dose through the first progression. We want to leave the patient on the therapeutic until the second progression. There's no added toxicity. The patient goes off Sutent. They go to standard of care, which are other TKIs that are used in the field. Eligibility criteria, again, 1 to 4 risk factors only. We want to eliminate the poorest risk. I don't think any therapy will benefit them. So we're looking at this point, about 75% of our patients are intermediate risk, 25% are poor risk, and we're about roughly 20% through the enrollment of these 450 patients. The primary endpoint, and this is clinically significant, would be a 6-month improvement in overall survival. You remember earlier we doubled overall survival, 30 months versus 15 for Sutent alone historically, but it's a small study, it was not controlled, so we took a very conservative but yet still clinically beneficial estimate of the improvement and powered the study at 80% to show that. So without going into too much detail on this, Arm A, it's a dose every 3 weeks times 5, and then quarterly dosing. And so there'd be 8 doses in 48 weeks. We would continue boosting the immune system at quarterly intervals to the second progression. Arm B will start out with Sutent, first-line therapy, and then move to other TKI or mTOR inhibitors. And again, the final endpoint will be overall survival. I have to move up. So here's our key milestones. Trial was activated. We have 140 sites, North America, Europe, and also Israel. We felt that number of sites is necessary to reach the 450 goal by the end of 2014. By 2015, we're going to have futility analysis at 25%, 50%, and 75%, and 100% of events. The number of events required to see a 6-month improvement in survival was about 290 with a 6-month improvement. If we had a 8-month improvement, we would observe a significant difference in overall survival at the 75% point. And fortunately, at this point in time, there's no competitive product enrolling in this patient population, which is very good for our enrollment. I don't have a lot of time, so I want to briefly come to AGS-04 for HIV. I want to mention the randomized, double-blinded, placebo-controlled trial. We've done 2 earlier studies than this. This study was different in terms of continued dosing for 12 months. The primary endpoint is to see greater than a 1.1 log, roughly a 10-plus log reduction in viral load after we interrupt the patient on antiretroviral therapy. So we interrupt the patient, continue dosing, and we monitor their viral load as well as their CD4 counts. We've completed enrollment. We're cleaning up the database. And positive data by the end of the second quarter would provide us with a proof of concept to really take the big step and aim our treatment more at the eradication of HIV. Again, fully funded by the NIH. Quickly on we plan 2 trials, the adult trial and the pediatric trial. In the adult trial, what we're trying to do is use latency reversing drugs like vorinostat, these HDAC inhibitors, to expose the virus to our immune therapy, and the endpoint would be to totally eliminate the virus. In the pediatric trial, where these young children have HIV and are on ART, they tend to be able to control the virus, but they have no antiviral memory T cells. So what we want to do there is use the vaccine and prevent the need to use these antiretroviral therapies. I only have 7 seconds left, so quickly on the automation end, we have 3 instruments that we built ourselves. One is a fully automated robotic instrument to make the RNA in 12 hours. The other 2, one's involved in producing the mature dendritic cell that we're going to electroporate the RNA into. And the final one is basically electroporation in a finished fill device. One facility can serve all America, North America. The cost of goods is going to be comparable to most biologics. We're talking about a 10% cost versus, I think, what you've seen for Dendreon, 50 to 60% cost of goods. What it really does is it lowers labor dramatically, decreases the size of the facility, provides consistency, because at 10,000 patients, I really don't believe human beings can do this process or any other complex autologous process consistently. And it's protected by U.S. patents, but primarily on the cellular devices, those are trade secret because many of the automated steps there would be applicable to other therapies. So this is our product pipeline. I already talked about the phase 3 study. We're starting studies in earlier stage renal cell carcinoma and other tumors, phase 2A studies, 2014. I already mentioned the studies that we're planning for pediatric and adult HIV populations. And then in summary, we've got the phase 3 study that we expect to file a BLA on in 2016, reaching our overall survival endpoints. On HIV, we have the studies I mentioned, along with unblinding, the current randomized double-blinded study. And then finally, takeaway messages. This therapy induces a cell that we know correlates with good clinical outcome. If we get it approved for renal cell, it's applicable to a variety of cancers, and we feel we've solved the manufacturing issue in terms of cost of goods, scalability, consistency, et cetera. And the finances that we raised during the IPO are sufficient to fund both this pivotal trial as well as the other oncology studies I mentioned. In HIV, where expectation is these newer studies will be partially, if not completely, funded by the NIH. With that, thank you. 2 minutes over.